Hello, and welcome to our next lecture here in cognitive neuroscience. We'll be finishing up our last two lectures in methods. This one will be an introduction to functional neuroimaging, mostly breaking down some of the methodology issues, introducing some of the concepts, and then in the next lecture, which will be the last in methods, we'll actually dive into positron emission tomography, or PET scans, as well as functional magnetic resonance imaging. So let's dive right in here. So here's a look at this overall lecture. I divided this into two because this was going to be too long to do in one spot. I don't want you having to sit here through listening to me talk for too long. So let's start with an introduction to functional neuroimaging and then talk about some of the experimental designs and some of those issues. We'll then finish up with these techniques and some limits of functional imaging in the next lecture. So let's start off with an introduction to functional neuroimaging. The goal here is to identify the physiological changes in specific regions of the brain. And so we're trying to determine where in the brain something's happening. And that's the goal throughout all of these techniques, is to try to both understand how events unfold in real time, which we do with the event-related potentials, and somewhat with functional magnetic resonance imaging we'll see here in a bit, but also localizing those functions to specific parts of the brain to try to build an overall model of how the brain processes information. And this is a really complex problem that is both reductionist, that is, we try to reduce it down to specific parts of the brain, but also very much not reductionist. That is, when we have to think about how all of this works together, as we start to tie all of this together, it's a pretty complicated picture. So as we move through the semester, we're going to be looking at each sort of individual component and then tying them together as we move our way up our way through the hierarchy of cognitive processes. So PET and fMRI, and PET is positron emission tomography, fMRI is functional magnetic resonance imaging. These do infer brain activity, but they don't directly measure brain activity. And that's what's really important. Because what we're doing is we're inferring brain activity through metabolic processes. So as we've talked in previous lectures, neurons are highly metabolic. They use a lot of glucose, they use a lot of oxygen, and as a result, when they're using those substances, we can actually try to measure that. So we're inferring functioning through these physiological processes. Glucose uptake and use of oxygen are the two primary measures we use. So remember, neurons have that high rate of metabolism. PET scans can measure glucose uptake. There's a specific radioligand that we can inject into a patient or into a participant that will allow us to then measure where the brain is using glucose by tracking glucose molecules. And it's a version of glucose, essentially a radioactive version of glucose. And so we can actually see where that glucose is being taken into a neuron, and that means that neuron has recently been active because of that glucose activity. Both PET and fMRI measure oxygen use, and we call this the hemodynamic response. And remember, one of the major functions of blood is to carry oxygen to tissues, particularly the brain. It uses 20% of blood volume. And so hemoglobin, which is what carries oxygen to tissues, either has oxygen, in which case it's oxygenated hemoglobin, or it does not, which is deoxygenated hemoglobin. And these two techniques measure that hemodynamic response in different ways. But essentially, it's that use of oxygen that allows us to infer where the brain might be busy at any given time. So the trick here is how to design these studies. And this is a quick overview. This isn't a how-to. There's a lot more involved in this. And so this is a brief overview of how this functions. So we're going to start with what's the dependent measure we tend to use here, because it is several steps away from what's actually measured. And so having a little bit of insight into that, I think, is really important. We'll then talk about blocked versus event-related designs. Block designs, all PET scans are block designs. There are some event-related fMRI designs. We'll talk about subtraction tasks, and then finally wrap up with trying to understand how these statistics are computed, and also understand what exactly it is that you're looking at when you look at a magnetic resonance imaging plot, like you might see on the news or in your textbook or many other areas that we're going to look at. So the first part off the bat is we have to introduce some new lingo, and that is, what is a voxel? And a voxel is a portmanteau of the words volumetric and pixel. And just a side note, portmanteau is one of my favorite words in the world because, I don't know, I just love it. So email is a portmanteau, for example, of electronic mail. So voxel is a volumetric pixel. So it's a volume element, and a pixel is these things you see on your screen you're looking at right now. So it's a volumetric element representing a value on a regular grid in a three-dimensional space. So when we're talking about voxel analysis, depends on the technique we're using and its sensitivity. So in general, when we're talking about something like functional magnetic resonance imaging, we might be talking about a three-dimensional cube, 3 millimeters by 3 millimeters by 3 millimeters, or maybe even a millimeter square. We're hoping to get down to 0.1 millimeters eventually with these newer magnetic resonance imaging techniques. But it's that 3D voxel that really is what we're looking at. And so within those voxels, we're looking at, is there a change in the signal from the magnetic resonance image machine, or from the signal from a PET scan? So ostensibly, our dependent measure might be changes in the BOLD response, which is the blood oxygen level dependent response from an fMRI, and we'll get into that in the next lecture, or other changes in the brain. So for example, with PET scans, it'll be a change in the detection of radiation from the glucose that is being taken up in the brain. There's a different technique used for oxygen. There are also other ways in which PET scans are used to look at density of receptors and other structural issues. We'll get into that in the next lecture, because here we start to get into the line between structure and function. So that's ostensibly what we're measuring. What we're really measuring is the signal returned to a PET scanner or an MRI scanner. And so as I said a minute ago, we're sort of several steps removed from what's measured versus what we're interested in. So we get a signal return to the PET scanner or an MRI scanner from a specific location in 3D space. Generally, we oftentimes want to examine regions of interest. And essentially, this came about originally because the technology was slow and data processing took too long. And so rather than imaging the entire brain and trying to crunch the numbers on an entire brain, we would look at a region of interest. And so we might look, for example, in the inferior parietal lobe when we're doing an attention task, because we know individuals with damage to the inferior parietal lobe have difficulties with attention. And so we might look at that region of interest. And really, this is how all of these studies should be done. They should all be hypothesis-driven. That is, they should all start with, we expect to see changes in part of the brain. Not to get too technical. So there should always be some kind of sort of region of interest. Now, we don't, in most newer studies, do regional analysis like we used to, because the technology is so much better, that the data processing is so much faster. It doesn't take an entire weekend to download data. So somebody who used to work with when I was in graduate school did fMRI research and would essentially start file transfer on Friday. It might be done when she got back to the office on Monday. It might not. So we've come a long way. And in fact, the technology is so good now that they've developed bedside MRI units that have very, very low magnetic resonance or magnetic fields, plugs into a basic outlet like you would have in your house, and they can image the brain of an individual without them having to leave their hospital bed. So we've really come a long way. So one of the things we'll often do is we'll test a voxel by voxel analysis. So we'll basically do a test of this voxel versus in one condition versus that same voxel in another condition, or we might look at this voxel versus a different one. So if we're looking at left versus right hemisphere, we might look at if there's a difference in the return rate of the signal from the left hemisphere to the right hemisphere in that specific location in the brain. It starts to get really difficult. So this is where we sort of start looking at what exactly it is that we're looking at as we're starting to do this analysis. The next thing we have to think about with experimental methods here are blocked versus event-related designs. So PET scans in many fMRI studies employ block designs. PET scans kind of have to because the scanning technology is not that fast. And one of the reasons why it's not that fast is because the precise nature of trying to measure PET scans has to do with the decay rate of the radionucleotides. So the way PET scans work is you're injected with a radioactive isotope. As that radioactive isotope decays, it gives off gamma radiation. That gamma radiation is then measured. And the faster the decay, the more the radiation exposure. And so we generally have to do blocked designs. That is, you do one thing for a while, something else for a while, something else for a while. In MRI studies, or fMRI studies, a little bit different. The technology is quick enough that they can scan quickly, particularly in areas of interest, while you're engaging in a particular task. So it's sort of blocked, but not as blocked, I guess I should say. So with a PET scan, you would have to do basically, particularly in the older scanners, you would have to do all of one task, then all of another task, and all of a third task. You couldn't randomly order those, and that alters the way a task is designed. So this can create some problems. So in the early PET scans of memory tasks, so one of the things we do a lot in memory studies is we use what is called recognition memory. And essentially you study a list of words, then you get a new list of words, and your task is to determine which words you are from the study list and which words are new lists or new items. The problem with PET scan is they have to then present blocks of old items and blocks of new items. Whereas in a basic behavioral study, let's say you studied 50 words, you get 100 test items that are in random order. So you get an old item, 2 new items, 3 old items, 1 new item, etc. Whereas in the block design, it's 10 old items, 10 new items. And that alters the behavioral response of the participant because it creates an expectancy. So, oh, that wasn't on the test list. Neither was that one. Well, neither was that one. And so they start to expect, so they stop using their memory and start just judging the task based on their expectancy more than their memory. So that creates some problems. The way we might do this in an fMRI study can be problematic, but less so because it's faster. So here what we can do is we can sort of move from one thing to the next and then make comparisons. So in this case, we have a blocked fMRI design looking at moving versus static random dots. And then we sort of have this over a time frame. So the person's sitting in the scanner, and we're measuring, this is the primary visual cortex. And the dots are moving, or they're static. And they're moving, and then they're static. And then there's breaks in between for you to fidget and breathe, and for the scanner to reorient itself. And then over here we have what's called area MT. And this is the area of the brain that's responsible for processing movement. So you can see when the dots are moving, lots of activity. When it's static, not much activity. When it's moving, there's activity. When it's static, there's not much activity. So this is a nice example of this kind of block back and forth design where we look at the overall analysis and signal change. And here we're looking across a region. This would be a region of interest study, not a voxel by voxel study. But what we're able to do then is we're able to compare static and moving dots in 2 different parts of the brain. And we use the static dots because we obviously need something engaged. If we're only interested in motion perception, we have to have something that's moving and something that's not moving, but that's doing everything else. Well, that's going to get at the subtraction method I'm going to talk about here in a minute. So, but what we can do then is we can see that movement creates a little bit greater signal in the primary visual cortex, probably because the dots are moving, it's activating different parts of what's called the retinotopic map. So it's not the movement per se, but just the fact that there's more stuff in more locations. Then we get to area MT, and we can see the static dots really aren't doing anything to activate that part of the brain, but area MT is getting activated by that movement. So that gets us to is a look at blocked fMRI studies. Now we're at the point the scanners are fast enough and our data analysis techniques are advanced enough that we can actually do event-related fMRI. And essentially what we're doing, this is a similar logic to event-related potentials. We time lock neural signaling to that external event and average over a number of trials. And essentially, we're able to get pretty close readings on the time relationship of events because we're signaling, or we're sampling parts of the brain at different times and getting responses to a stimulus at a specific region across different times. So you start the scan and the stimulus sort of in different relationship to one another. And what you're able to do then is say, okay, we're scanning this voxel 10 milliseconds after the stimulus onset. Then in another trial, we're scanning that same voxel 20 milliseconds after stimulus onset. And then a third scan, 30 milliseconds, etc. So it's basically simply altering the computer programming, altering the sampling technique, altering the way we examine the data. And so what we're able to do then, just like before, we have stimulus presentations, and we're able to examine that blood oxygen level dependent response across time, and then try to get some average of what's happening across these time frames. So the final note on both fMRI, not final note, sorry. One of the notes on fMRI and PET scans is we do have to have some kind of subtraction task. So, like most experiments, we have sort of a control and experimental condition. And this is a little bit more complicated in PET and fMRI studies because, just like with ERPs, the brain's busy doing a lot of other things. So we can't just scan the brain and see what happens when we present a face, for example. We're interested in face perception, and all we do is measure what happens when the brain looks at faces. We're going to get a bunch of other stuff. We're going to get just basic visual processing because we're looking at things. We're going to get breathing, hearing, because MRI machines are really loud. So the whole brain's going to be lit up like a Christmas tree, and it's not going to tell us anything about face perception. So what we need to do is we need to subtract out all that noise. So in ERPs, we're able to average out that noise. But MRI studies are so different because of the nature of the data that the sound of that MRI machine, the visual processing centers, all those things are going to be returning signals. But what we're interested in is face processing or motion perception. So we have to think about that. So that gets us back to motion and visual perception. So when we start trying to generate those fMRI maps that you see, where you see a brain and it's all lit up in some area, and we're interested in areas involved in movement, well, what we're going to do then is we're going to take all these movement trials and all these stationary trials, and we're going to subtract one from the other. So what we're going to do is, because we're interested in movement, we're going to take the movement and we're going to subtract the static trials. What that's going to do is we're going to get nothing in the primary visual cortex. It's involved in motion, but it's also involved in just static perception. So it's not involved in the perception of motion. It's just involved in visual perception. by subtracting out the stationary trials from the motion trials, we get to see just what is only associated with motion. So then when we come over here to area MT here on the right, we subtract these stationary trials from the movement trials. We're going to see a big signal because there's nothing happening in those stationary trials, but there's a big signal associated with those movement trials. And that's the logic behind fMRI research. So face perception, and we're going to get to this later in the semester, is a whole other problem because this turns out there's really nothing like faces. And so there's a bunch of different things people try, and so there's enough studies involved now that we basically know what parts of the brain are involved in faces. But here we have faces versus objects. So again, you can see the face trials versus the object trials in some part of the brain. And then here we have intact faces versus scrambled faces. This is probably a little bit better because it's all the same features, but they're not assembled in a face. And so we can look at scrambled versus intact faces and see what part of the brain is involved in that assembling these perceptual features into a face and seeing a face. And that's important for understanding face perception, but also, so let's say we're really interested in trying to come up with the best camouflage for the military possible. How do we camouflage a face to the greatest extent possible? Well, we can try out a bunch of different camouflage. We can obviously do behavioral techniques, but we can also look at what kind of return we're getting from the fusiform face gyrus in this kind of study. So lots of interesting stuff being done here. That gets us to some statistical considerations. Now, we talked already about the enormous amount of data in event-related potential studies. Here, it's gargantuan. I mean, this is an enormous amount of data. And that requires a lot of careful consideration, but also pretty significant data correction. So if you remember, some of you might not have taken statistics yet. One of the things in statistics is we try to limit our error rate. And so essentially, if you think about if we ran an experiment randomly 100 times, 5 times, those statistics are going to come out significant by sheer random chance. And that's related to what we call our alpha level. The problem is, in an fMRI study, we might be talking about 100 million comparisons. So that means we now have 5 million significant results by random chance. So we have to really alter the way we conduct studies. We do lots of data correction. There's all these corrections to account for the assumptions of statistics, which are way beyond what we need to talk about here. But it does require a lot of massaging of the data, as we like to say. The other thing I want to talk about is what you're actually seeing. So it's not very often you see those graphs we've been looking at. Usually what you see is something like a statistical parametric map. And these are presented on a standardized brain. So when you're looking at an fMRI study, you're not looking at the results from any person, and you're not looking at anybody's brain. You're looking at an average brain that's been created from scans of 1,000 people. And the data is then projected onto that sort of standardized or idealized brain. And that's really important for you to understand because these are averages across numbers of participants. So it's 25 participants, and their data, or the difference between these 12 people and those 12 people, and their differences in brain activity because this group's ADHD and that group's not. But essentially what we're looking at is a statistical parametric map that is put onto a brain. So this is what you're seeing. So these statistical parametric maps are used to present the results of the statistical analysis. So voxels, those 3D volumetric spaces, whose p-values are below a certain threshold, and those p-values are an important part of statistics. So when a p-value is below that threshold, we call it a significant difference. All you need to know about that. And these are color-coded to signify that they contain significant task-related signal differences. And so essentially what we're doing is we're statistically testing the signal return in those two areas, or in that area from these two different tasks or across two different people. And so in that voxel where we get this p-value below a certain threshold, that is a certain level of significance, that's when we get those maps. And so usually they're sort of yellow, orange, and red, depending on what that level of difference is between those two groups of voxels. And those p-values, the greater the difference is between those, the lower that p-value is going to be. I know, this is a lot to take in. We'll take a breath. So this is what you see. So this is a caffeine versus placebo. I have no idea. I just yanked this off the internet to show you a statistical parametric map. I have no idea what they were doing here. But you can see they've done this two ways. They've looked at, they've put the statistical parametric map with these different values across these specific areas. And here we have the dorsal putamen and the ventral striatum. But we're going to be looking at these kinds of maps. These can be placed in slices. They can be placed on the surface. So essentially this idealized map is what you're looking at. And that's really important for you to understand. I do want to make a final note about this kind of data. There is emerging evidence that this data is more persuasive to people than other types of data because of TV and movies and whatnot. This data is no more important than any other data. It's just what people think is important. And so there's a lot of hype around this kind of data, but data is data as far as I'm concerned. And so all of these techniques have value. Behavioral science has value without any neural imaging. All of these are used to conduct these investigations. So that's what I want to finish with. We're going to pick up on functional neural imaging, specifically PET scans and fMRI scans, and some more details about those in the next lecture.